types of network methods that I've worked on for brain imaging that I want to talk about. First, give acknowledgments to the two different groups of people that are working on these have been at Warwick, and some of them actually have moved on. Lily has gone back to Brazil, and Anna has moved on to National University of Singapore, and those are the two main people that are responsible for this kind of work. So the two different pieces I want to work on, one is actually astutely and exactly trying to respect the dynamic nature of the functional magnetic resonance data that we're working with. And the other one is kind of trying to dip my toe into this sort of graph analysis world or graph theory world that brain imaging people like to do, and see if we can improve what they're doing by using some stochastic I would say some models that are based with some statistical principles. So in brain imaging nowadays, everyone is talking about the connectome. The connectome is just a fancy way to say we want to understand how do different parts of the brain connect to one another. Ideally, we like to sort of know about directed influences, but usually we just suffice it to work with correlations between different brain regions, and then then people try to cluster and try to understand what's the sort of full correlation or partial correlation. A lot of times they just focus on the full correlation, even though I think we've heard about partial correlation that might be more insightful. What I'm going to talk about is actually trying to use this time series information to learn the directed structure between different brain regions. But to start there, I need to talk about this dynamic regression model. This is a sort of a standard thing in Bayesian circles of a Bayesian model. It's well known to me and my colleagues. Jeff Harrison was the founding department chair of the University of Warwick's Department of Statistics, and sort of this is kind of in the DNA of the department. I certainly knew nothing about it until I went there. So it's a state space model, but you can also think of it as a regression model that we have regressors, and this is kind of a standard fMRI setting. So we have data over time, we have regressors over time, but what's different, of course, is that these regression coefficients are time varying, and they evolve as per this state equation. And of course, as you write it just right here, this seems completely unidentifiable. How would we ever know what's going on? And of course, we need to sort of have some assumptions on the variability of the state parameters. And the trick that gets used a lot with these models is variance discounting. So if we assume that the variability of the state parameters is yoked, is a multiple of the observation variance, suddenly things get a lot simpler. And in particular, if we can go and we can estimate this, it turns out all of the equations actually have a much simpler form, and we can actually write down a closed form posterior with no sampling, nothing required, conditional on this discounting parameter. And so if the discount, so you can see if delta is 0 here, then this is static, and we actually have a regular regression model. There's nothing dynamic about it. And if it's a half, it kind of looks like a random walk. And you can interpret this delta parameter as kind of the information lost. 1 minus delta is the information lost at each time point t. So just graphically, so that's just a regular dynamic regression model. It's used as sort of kind of a standard Bayesian time series model. I'm interested in, can we reconstruct the graphs of some small number of networks, small number of nodes. So here's, as a starting point, the standard structural equation model can be thought of as like this, that at each point we have data that's being driven by here just some white noise source, and of course I'm illustrating that there's nothing linking this over time. We need the repetition somehow over time, over subjects to estimate this, but there's no sort of link. Everything is assumed to be independent. This node here actually is just the white noise. This node here is actually a sum of some white noise source and some fraction of this data, and this node here is a sum of some white noise and a fraction of all these. But all this is static. That's a standard structural equation model. The model that we're going to entertain is the multi-regression dynamic model, which actually says, okay, actually we want these things to be time varying. Now sometimes when you see dynamic Bayes nets models, you'll have that the dependence is in the past. Like this node here would be dependent on the past of some other neighbor. And that's not. This is all driven by instantaneous correlations, the dependence. So this is saying here, this node here is now sum of white noise and some sort of constrained, drifty, slowly varying intercept. This model here has got the white noise, some sort of constrained intercept, and then some time varying dependence on the other node. Here, this has two parents as well as this intercept parameter. So that's the multi-regression dynamic model. Had been applied in sort of traffic modeling settings, and the idea is we want to try to learn what the structure is just based on time series, on a multivariate time series data set. So I kind of want to spend, I think, at least today, there's been a lot more about sort of large-scale graphs, so I'm kind of going to race through this first material to get to the second. But just briefly, you can write the model at each node R as a linear combination of this F matrix. This F matrix has an intercept, and then the data from the parents. The node has some parents, and the data from those parents is in there. So it's saying I'm a linear combination of an intercept and my parents and some white noise. And then this is a system equation for these variables that control my dependence on my parents and that intercept. And then here we go. Here's a statement about the variance discounting. I'm saying that the variance of the state equations are a multiple of this observation variance. I've written this variance as time varying. In practice, we assume that the observation variance is constant over time, which seems like a reasonable assumption. And again, the real sort of magic here is that if we use maximum likelihood to estimate delta, everything is conjugate. It's amazing. So the posterior of the regression coefficients theta, that is a multivariate non-central t distribution. The filtering posterior also is a multivariate t. The predictive distribution at time t. And crucially, the model evidence, the log predictive likelihood that we use to compare different models and get Bayes factors, also closed form, and it factors, it sums over time, and it sums over nodes. So this is a really sort of computationally efficient and advantageous thing. So that's great. It's a nice Bayesian model, but no sampling required. So this it was designed to find directed acyclic graphs. And now, of course, searching model DAG space is a problem because it grows exponentially. I learned about this online encyclopedia of integer sequences. That's such a cool thing to browse around and look at sequences of integers. Anyway, you can see it blows up. Just even for 7 nodes, 58 billion different DAGs, an exhaustive approach does not work. And there have been a number of approximate methods that people have used. Peter Spirides and Clark Gleamore have been involved in these. And then we're sort of shocked to learn that the people in the computer science community have been taking a different approach, an integer programming algorithm, because our likelihood is just a sum, additive term here. You can use these approach, and then suddenly, instead of sort of this exponential explosion, actually we only need to evaluate all the different configurations of the model at each node, and so now we only have n times 2 to the n minus 1 evaluations. This still is exponential, it grows up, but it doesn't grow up this badly. So we're able to do things, the slow step is actually evaluating all the different, basically for each node I have to say what are all the possible configurations of my parents. There are 2 to the n minus 1 possible parents I can or cannot have, and that's what I have to consider. And I just have to do that for each node, and then the actual running the IPA, the integer programming algorithm, is really quick. So we wanted to evaluate this method with some data, but of course with real brain data we don't know what the network is, it looks like. Now an excellent resource if you're playing around in this area is this NetSim paper. This is by Steve Smith and all. Actually I'm a co-author on the paper where he considered a bunch of different networks, 5-node, 25 and 50-node networks, where he used a realistic generative model for the fMRI data, where we, of course, we know the truth. And he also considered a number of different simulations messing things up in different ways. And so the one that I was interested that he'd mess things up is he messed it up by changing the strength of the connectivity over time, which of course is what this model is designed for. So we took this simulation, you can download it, and he has 50 realizations, and we wanted to see how good are we at recovering just the network structure at all, ignoring direction, and then also recovering the directionality. So we compared the other methods that he looked at, this GES and PC algorithms. These are kind of standard methods in the BayesNet world. And if you look at the sensitivity, specificity, PPV, and MPV, all these things to try to say how well are we finding the edges, we're doing a good job. We're not always the best. We're here at MDM. We're not always the best one. But when you consider that these models here are static models and they don't have this extra flexibility, and we are allowing for that flexibility, I think it's pretty impressive that we're pretty much in line with those guys, even though those ones ignore the dynamicness. And then in terms of directionality, we're also doing well, getting about just over 60% recovery, which is better than all but their best method that they found in that evaluation. So people are always very cautious and for good reasons about recovering directionality in real data, and we seem to be, at least under simulations, we seem to be doing this well. I actually don't have this slide here, but I've also looked at trying to evaluate problems with the hemodynamic response. So I've actually written a paper saying people should never do Granger causality on fMRI data, and we've done another simulation to sort of address that, but I don't have a slide for that. You do okay, just a take-home. So what I've just shown is that this seems to work okay under simulations, and it can recover DAGs. The problem is, as soon as I presented this to my neuroscience colleagues, they said, the brain's not a DAG. What are you thinking? There are reciprocal connections all over the place. And so Jim Smith, the guy who's worked on this methodology, said, don't worry. Actually, what you should do is actually just forget the constraint on the optimization that says you must find a DAG. The log predictive likelihood is a sum of terms. Just optimize each node individually. So properly, we don't actually fit a statistical model. If there are regions, we're fitting our multivariate models, and they may not be consistent in a statistical way, but as we see, they're actually more consistent with the data. They behave in a decent way, and we're calling this a dynamic graphical model. So we applied this to 32 subjects worth of fMRI data. These are sort of nice high-speed acquisitions with 1.1 seconds in between each sample. And now, of course, the problem is these are not large scale. I cannot do 100 regions with this, but this particular study was interested in 12 regions that are actually involved in pain. This is actually a resting state study. No one was hurt doing this task, but they used this to compare to later stuff where they actually did hurt people. So we're going to try to figure out which of these 12 nodes are connected to each other. So if we first look at just partial correlation. So here is partial correlation. And what I'm doing here is it's sort of a funny thing. I'm just looking at how I have to fit this model each subject individually, and I'm just looking at how common are our edges across subjects, and I'm going to look at just which ones occur significantly. That is, I'm going to say if there are, say, on average across all subjects, if there are 20 edges, I'm going to do a binomial test to say if those 20 edges are even uniformly spread over the whole network, how likely is it that I've seen an unlikely large, consistent proportion of edges at one particular edge? So this is basically, we're doing a binomial test under a null hypothesis that there's no consistency of any of the edges across any subjects. So what we're seeing here is just a pattern of significant edges across, significantly prevalent edges across the subjects. It's not a multi-subject model yet. And so, of course, what you see is partial correlation is that you have symmetric, and what we actually get to see here is that we have this evidence of directed, so that the orbital frontal and dorsolateral prefrontal regions are parents of children in the anterior cingulate, and it doesn't go the other way. These anterior cingulate, these posterior regions are not parents of these frontal regions, which, when we talked to our neuroscience collaborators, said that this was very reasonable. And I should say that this, I didn't even show you the DAG results because I wanted to save time, but this result actually doesn't work at all because you can see there are lots of reciprocal connections. The left and right anterior insula are connected. The left and right posterior insula are connected. And those things would just not be possible if we're doing the proper DAG approach. And so when we did this map, when we turn that connection matrix into something graphical, this is basically just saying there's this tendency of these frontal regions to project back, but not vice versa, but all the posterior things are sort of interconnected and the frontal things are interconnected. And again, we don't know ground truth, but our neuroscience collaborator said this is a reasonable thing, and sort of matches what they understood about the brain, and in particular this frontal region sort of being a driving region. So the things that I'm going to do with this going forward, just at the human brain mapping meeting, we met a group that was doing this same sort of thing, but on mice. So collecting resting state data on mice, and in mice we actually have some ground truth information about directed networks, because the Allen Brain Institute has done these exhaustive studies where they put a viral tracer in one neuron and see where that tracer ends up when it goes up the axon. So this is really exciting, so that we're actually going to have some ground truth to compare our directed networks to and see if we're getting sort of reasonable results that match that. Okay, so that's the end of the first part of this talk. I was drawn to this work because people are trying to understand directionality. This model is computationally efficient for this in this regime. And now what I have not talked about, I didn't really actually show you any dynamic plots, dynamic time series, because so far while we've got an excellent or a very interpretable network structure out, resting state are just sort of randomly changing covariates, and we haven't sort of made any sense. We are looking at ways of trying to fit this to data that has structure or task to it, and see if there's some consistency. Actually, one thing was recommended us was to take Jack Gallant's type of data, where he shows movies, and then see if we show the same movie to different subjects, do we get similar patterns of connectivity over time across subjects. So that's something we want to do. And the last thing I didn't mention is actually there's also some ways that we can even make this even faster if instead of doing all possible models, consider all possible models as just doing a greedy network search, and that could actually push us up to 20, 25 nodes in this sort of setting. Okay, oh good, 15 minutes exactly. So now if I look at, and now I'm going to step towards the type of networks I think that we've been talking about earlier in the day, and looking at large-scale networks. If you talk to anyone in brain imaging, they'll often talk about graph theory, and they're very interested in using summary statistics of graphs and relating them to fundamental properties of the brain. And this stuff always gives me pause because it always seems like quite a leap to go from sort of, how expensive is wiring to, what the brain is doing. So when I got a student shared with Ed Bullmore, who's here in Cambridge, I thought, okay, well, I'm going to try to take all I know or all I can find out about sort of stochastic models for graphs and try to put them to work in his setting and see if they, if I can change the way he thinks about these things. So the first model I found was something called the Erdős-Rényi mixture model. It's nothing more than a stochastic block model, but it's a particular implementation. It uses EM on a variational approximation, like Peter was talking about just before lunch. And then also really handy is that it uses something called an integration classification likelihood to estimate the number of nodes, a number of blocks. So it's a handy, efficient package. And certainly we did learn quickly you have to do many, many random restarts to actually get yourself to a global optimum. But we weren't doing gigantic networks, as you'll see. And so this was just a handy So we started out by saying, okay, this looks like a reasonable tool. Can we validate it and show that it does sensible things in brain networks? And even though I'm interested in humans, actually Ed convinced me that we should start with the worm, because again, with the worm, we know what each one of these neurons are doing. We actually know a ton of things about them. We know which ganglion they're organized into, what kind of neuron they are, neuron class, are they doing locomotion, where are they anatomically in the worm, etc. So our idea was, like, let's apply this stochastic block model and then compared to what at the time was sort of what everyone seemed to be doing, which was modularity-based community estimation. And so we wanted to compare these, and then we can actually do things like we can basically use interclass correlation or the adjusted random index to compare the estimated blocks that we get with the categorical measures and ICC to measure how well are those blocks, the estimated blocks, explaining the quantitative variables. So here's the estimated structure of the 350-some neurons of the C. elegans. And the first thing is that our collaborators said, oh, this is great. These are hubs. This is exactly what we expect to find. And crucially, you also find this highly non-modular. So these two things look like they go together, but in fact, group 2 is very different from group 1 in terms of its connection to group 8. And of course, if you just throw a modularity-based method, it just really can't sort of get that sort of non-modular structure. But we can now quantify that. And across all these different things, whether we're looking at the adjusted RAND index, there's exactly one where this stochastic block model doesn't do as well. That's for the neuron type. But in all the other methods, this block model is outperforming the modularity-based methods. So basically saying that, okay, before we step to the human, this thing is actually doing something. It's doing something that's capturing biologically meaningful structure in the network. Okay, but the other reality, if you're going to work with humans, is of course that we have more than one. We want to try to generalize these inferences across subjects, and also, as Peter stressed, we usually have covariates. And so you can't do a brain image analysis without adding a covariate for gender and sex, for example. So if we just applied this black box method, of course we're going to get different networks for different subjects, and they wouldn't align. So we wanted to see how could we make comparisons, make inference, and use covariates. So we extended this model. So here's the basic ERMM stochastic block model, and extend that in a number of ways. We can consider what we call a binomial mixture model, where we just make the assumption that these groups are shared across subjects, and then the counts, in essence, are binomials at each edge. A homogeneous Bernoulli mixture model, which now says that the incidence of edges basically varies by a global factor across all nodes. Now, this could be useful if we are worried about kind of just bias in the way that we create these binary networks, And so, for example, if you have subjects that's 20, 40, or 90 years old, it could be that with age the data just degrades in quality and that the overall prevalence of edges go down, and this model would explain for that. But perhaps the most useful model, and the one that we did indeed end up focusing on, is one where we basically just have this regression model at each block, so that we do not assume any particular structure across the different blocks, but again, crucially, the block structure is common across all subjects. So this is basically like, because we are assuming independence within each one of these blocks, it's this basically like a logistic regression model, an intersubject logistic regression model. So a minor point is that we do have to account for, if you've ever done logistic regression, there's this problem called total separation, where if the covariates line up with your observation, so basically if you have a small amount of binary data and you had a covariate that was male and female, and if all your males were false and all your females were true, basically you can drive the likelihood to infinity by because you can make a perfect separation between the two. And also, there's this thing called Firth regression, named after David Firth in our department, which is basically saying if you use, he uses a likelihood, a penalized likelihood motivation, but it basically is a Bayesian approach where you use a Jeffreys prior to say on the parameters, and that addition to the likelihood regularizes this, and you don't actually have problems with the total separation. We then set about developing inference procedures conditional on the block assignments of of inference on these beta coefficients with a walled test, also comparing that to a permutation test. So I'll just show you the null hypothesis simulations to show that we are doing something reasonable. It's a very small number of, here Q is 3, but we did consider network sizes of 5, 100, or 500. This is kind of the null case. There's equal prevalence in each, so there is no structure here. You will never be able to recover this. There's no covariates in this simple case either. And then we considered different kind of network structures. And then this one is also very punishing, 0.3, 0.31, 0.3, 0.29. So this is also a very punishing case. And we consider balanced, mildly unbalanced, and severely unbalanced cases. And oh, I'm sorry, actually I forgot, there is a covariate here. There's an age effect, and that's the effect that we're going to be testing for. We're going to be testing for, we're going to do a walled test for the age effect. And so just showing you the null hypothesis, false positive rate control, you see that on these punishing cases, P1 and P5, as the number of nodes grows from 50, 100 to 500, well, they just never work. Because there is basically here, there's no structure to identify. But in all the other cases, we are getting, even with few as 50 subjects, we're getting false positive control in the expected area, except for these really tough networks. What's the other one here? P2. This is also very hard here because these two are basically these are indistinguishable basically. These are close to indistinguishable because of the similarity there. But with enough data, all but the P1 and P5, we're getting the nominal false positive rate, and we also computed the power as well. But I'm not showing it. So to show this in an application, so now a different data set. This is 25 subjects. We were given 297 nodes per subject. Ed Bullmore is fond of doing a wavelet transform to his data and then correlating the wavelet coefficients. And then we created binary networks from this after Fisher transform of the correlation coefficients and FDR thresholding at 5%. The covariates that we have of interest is that the control and patients. So this is schizophrenic patients and controls, and we're going to model that difference, their age. The NART IQ, that's an estimate of the pre-morbid IQ, basically what would the IQ be of these schizophrenic individuals before they got sick. And also, because we're often worried about this in brain imaging, how much they moved in the scanner. So we estimated, the method estimated 22 blocks, and these actually sort of arrange themselves into sensible sort of networks. So we've kind of just manually sorted them just to show that these corresponded to networks that we expected to see, but also for any given color, they also kind of are doing sensible things. They're not sort of randomly scattered around the brain. And now we can, so here's just the reorganized adjacency matrix. Again, the key thing is that the structure, this network, the block assignments are common across subject, and now we can actually make inference on these, and I'll just whip through this. So these are now images of test statistics. Here, this is the z-statistic for patients versus control, and this is the z-statistic for the age effect being different from 0. And what you see, this is all negative. This is actually indicating that the controls have way more edges in general, except in some areas, than patients. And these have been FDR thresholded. The age effect is less prevalent, but again, also negative, and this sort of, well, maybe makes sense. The older you are, the fewer connections you have across the network. Okay, so I'm almost out of time. I just want to say some of the new directions we're pursuing with this. The people that do graph theory are very interested in one measure, which is called rich block. And so we're developing a method of rich block that basically implements this, we're using the stochastic block model to implement a version of that measure. And here, if you know rich club at all, they usually plot the coefficients. It always generally increases with degree. And these colored lines here are the approximate versions that we get with our rich block model. And we can also estimate the rich club coefficients. And we find that we get similar estimates of those coefficients as that the number of regions and number of nodes and number of blocks grows. And the other thing that's really cool, using some of the work that Patrick was showing, and hopefully we're going to do more work together, using his and Sophia's method of network histogram, one of the things that takes a long time when you do rich club is that you have to generate null samples. You have to generate sample networks that are drawn from the null distribution, and you do that over and over again. And that can be quite computationally intensive, especially with a big network. But if you have a model, you can just generate realizations from the simple sort of Bernoulli model. And so we've used the fitted network histogram model to actually do this rich club analysis on a 30,000 by 30,000 dense connectome, and then we've been able to identify these nodes as being rich in the rich club sense. So this is ongoing work. We just presented it at the Human Brain Mapping meeting last week or 2 weeks ago. Okay, so I've mainly been talking about connectomics. I guess the things that I find looking at sort of graph methods and what needs to be considered if you're going to apply any method to brain image data is that, well, so first of all, and this is probably seems like old news, you need to look beyond modularity. There's lots of non-modular structure that's interesting. You have to account for that. The brain imaging people need practical implementations. They need software that does it. And I applaud Patrick for when he published his paper, you could look on GitHub and get the MATLAB toolbox, and that's what my student used. I mean, I wouldn't have the time to go and implement it if I just had to start reading the archive paper. You need to accommodate multiple subjects and covariates, and as I've tried to demonstrate, ample validation to convince your collaborators that these things are making sense. So I'll stop there. Thanks for your attention.
Questions to come?
Could you say again what was DK in your covariates model? How is it related to covariates?
So DK are your covariates. So for us, this was age, gender, IQ, and your degree of movement.
But don't they depend on the node? So you have one for each node?
No, because that's the subject. This is your age. The age of the individual is the same for all nodes. I mean, I guess if you want to be really, you could work out a spatially varying degree to which a part of the brain moved. That would be something, but yeah.
There would be network sort of covariates, network level covariates, not node level.
Yeah, we haven't considered that because these are the things that people usually want to account for. I mean, that's another interesting thing. It'd be interesting to also have node or edge covariates, but I think this fits into a way I think a lot of scientists approach their data.
The second question: how did you fit? Is it easy to? Was it easy to fit?
No, this was a nightmare. This was like 6 months of my student's last part of her thesis. It was
variational.
So she's also using variational approximation on top of with EM, and it was just it was difficult. So the paper is still in revision. So yeah, it was not fun.
I have a question actually. On the first half, so comparing this dynamic Bayesian model with partial correlations, whatever else. Do you have an idea of kind of quantitative endpoint of analysis that one could use to say, at least in some particular context, this is better or this is more informative?
Well, what I like about this approach is that it is a regression model. So it actually does give you for each, so it's not just correlation.
You want to convince people that you don't want to use correlations to represent the connectome.
Yeah, so the way to convince them would be to, I guess, maybe integrate other external covariates. I mean, so far we've only done, brain regions on brain regions, but we could also include other covariates that made sense to be time varying. It's a good point. But I agree, there's more to convince people that it's the right model.
Let's come again.